That's it. You are now broadcasting. No pressure, of course. It's kind of hidden, so people can't see it just yet. But the idea is that this, whatever you are doing here, will be actually on the YouTube, and I'll check it later on.
So you live now.
So no pressure at all, but just very briefly, write big, so you can see. That's the eraser tool. That's your typing tool. The thing with the one, you actually have to hit where you want it to go first, and then type there, and then deselect is just press them. If you want to do any arrows or things like that, you're very welcome to. And these are quite nice. You can show little things like that if you want to.
Yeah.
Do you use a Mac at all?
Never before in my life, but I'll get used to it.
That'd be fine. Mark will help you.
Right.
Okay, so I think, if I just go back to here, and that's the microphones, I think we're all on. Device settings. Yeah, it's recording. Yeah, picking up. Good, good.
Lowy.
Good. Yeah, don't worry, that's not, it's just screen sharing. Don't worry. This country. Right. We'll get there. So that is only 15 minutes, 20 minutes to set up 2 rooms.
That's all right.
See, Mark, you're watching. You're going to do it.
Really? You're kidding.
Right. Nice.
20 plus.
Right. Yeah, get through it, which is fine. Cool. As always, we start off with a diagram. It seems like every paper starts off with a diagram of some sort, or label something on this diagram. So yeah, they've actually given us, rather than a little drawn diagram, little cartoon drawings, we've actually got an actual photo here. Not in colour, obviously, because they can't afford to print everyone in colour, but not that special. So, okay, with what they're pointing at here, it's a little bit grainy, isn't it? And it's kind of especially difficult because they haven't really given us the whole cell. We're just sort of seeing this tiny slice of it, so it's hard to tell what part of it we're looking at, what kind of cell is this. All we're told is it's part of an animal cell. Given that this is in an AS paper, we can expect that they'll be asking us to point out things that we know about and that we've learned about. So I would say, well, they give us, another thing to mention, they do give us a few sort of pointers to kind of get kind of our grasp. So it tells us that this here is the cell surface membrane. So we know that this is the edge of the cell, which is, helps us get our bearings. It tells us that this is a pair of centrioles because it gives us a question on centrioles in a minute. Easiest one to start off with would be B, I would say, because you can sort of see the inner sort of membrane here, and there's only really one organelle that looks that way, which is the mitochondria. They give you a little bit of leeway in what exactly you give the answer as, because you might have said this is, that's a mitochondrion, or you might say, that's the mitochondria's membrane it's pointing to. Generally, when it's not a drawing and it is a kind of grainy photograph like this, or micrograph, should I say, there'll be a little lenient, so you don't have to be really specific in what exact part they're pointing to, as long as you know what general structure it is they're asking about. So A is the nucleus, or you could have said the nuclear membrane or the nucleolus. Probably the most difficult one there, would you agree, to tell what that is? I mean, it sort of doesn't really look like you imagine the nucleus looks like, does it?
Probably because of the size, so it's like the main thing. Yeah,
and then you can't see all of it, and so you don't, you sort of picture a nucleus as this little black blob, and it doesn't look much like that. I suppose a few little, it's not really much of a hint as to sort of help you. We're going to get the pen as well. To help you sort of figure out what this is. You can, if you look really closely, just here, you can see a small opening in the membrane there, for where your ribosomes come out and things like that. So you might have picked up on that. But yeah, generally speaking, a very large black mass in an animal cell is usually going to be the nucleus. There's not really much else that it's going to be. So, and then C points to down here. Now this isn't really a pen sort of having a moment. This isn't really an organelle, so to speak, but this is more of a kind of space, and you can kind of see it's almost like a void in the rest of the stuff you've got here in the cytoplasm. You can see all, all the sort of bits floating around, which is kind of a hint to say that this is a lysosome or a vesicle. It's a small little sort of container full of proteins and other things that the cell has made that it's going to be traveling towards. You can see one here, moving towards the cell membrane, where it will leave the cell and empty its contents to wherever it needs to go. So yeah, that's A, B, and C. Okay with that? Yeah. Part B says, name one structure visible in figure 1.1 that would also be present in a prokaryotic cell. So there's a wide number of things that you could have said here. Basically, we're seeing if we know the difference between eukaryotic cells and prokaryotic cells, so an animal cell and a bacterium, maybe. So yeah, there's all kind of, and the important thing is you don't have to point out where it is on here. So although it says name a structure visible from here, you don't have to be able to recognize it from this picture. I mean, you do kind of see most of what you'd expect to see in the cell in this picture. So yeah, you could have said quite easily the membrane. They give it to you here. It should be like doodling on here. So there's the easiest answer is that is awful. I'm gonna have to get used to this. Yeah, this would be the easiest one because they've given that to you, and pretty much all cells are going to have some kind of membrane. So that would have been, I would say, the easiest answer there for B. But you could have said other things. You could have said the vesicles that we pointed out in C, or the cytoplasm itself. So yeah, if we're all right with that, we'll go on to part C, which is a slightly longer question, testing what we know about mitosis, specifically describing the roles of the centrioles. So you've got a few sort of points that we want to pick up on here. It's only a 2-mark question, so we're not worried about going too in depth about where things take place, but we want to hit a few key points, specifically the main role that these centrioles are playing, which is that they form these spindle fibers, these microtubules. And so you can pick up 1 mark there for mentioning just that they form these and what those are used for, so that they are used to pull the chromosomes apart during the anaphase of mitosis. And the other mark you want to pick up is kind of in the other finer details. So the fact that these centrioles will move to the poles of the cell, so that they organize everything and pull everything apart properly. But other than that, what do you think? Not too bad a question. It's only 2 marks, so we don't need loads and loads of detail there. It's not such a big deal. And that's all right. Is there a part? Yeah, there's part D. So this is the last part of this question. This is a 3-mark with a lot of space, so you might be tempted to just write. But it's similar kind of technique to the last one, as long as we get some of the key points down. I would say always, when you see a question like this, pay attention to how many marks it's worth, because if it's only 3 marks, you don't want to write 6 marks worth of answer, because we'll just be wasting our time and there's not really much point. So sodium ions cross cell surface membranes using facilitated diffusion or active transport. Explain why sodium ions cross cell surface membranes by these mechanisms and not by simple diffusion. So we're going to pick up a mark for explaining why they might use facilitated diffusion. We'll pick up a mark for why they might use active transport. And the last mark in this 3 will be why we can't just use simple diffusion. So you can kind of, that's an awful underline, it's sort of where it's at an angle. So we can kind of pick up from the question where it's worded where those marks are going to be and what things we actually need to talk about. So are we okay? Do you want to go into depth on this, or are we generally okay with this, or should we be able to talk about it? We go into it. So we'll start with why these sodium ions aren't crossing the membrane just by simple diffusion. So simple diffusion being a general movement of generally small particles. If there's a low concentration of them over here, they'll all diffuse this way. Note that it says sodium ions, which tells you that these particles are charged, and they won't be able to cross this self-surface membrane just on their own because they have this charge. Because the way that I'll try and do like a little drawing here. The way if the membrane looks like this, these heads themselves on this membrane. If you imagine you've got a lot of these like this, these heads are going to have a charge themselves. So if you have a lot of little particles come along that are all, say, positively charged, they're not going to pass this barrier because the charges are going to repel. So we need some other way to get them through, which is where facilitated diffusion comes in, which is it is a process that we're not using any energy here, but rather in this membrane we'll have some kind of channel protein. I'm sure they don't really look like this, but you can get the idea, which kind of acts as almost like a doorway for these charged particles that can't get through the wall like other smaller particles might be able to. They'll be able to pass through these channels, which might open or close depending on whether the cell needs these particles moving around. And so that's the second point there. That's facilitated diffusion. Final point being active transport. Keyword here is active, which is telling us that there's some kind of energy being spent here. Why might we need energy to be spent? If we can get them through these channel proteins, why would we need to spend any energy doing it if they'll just do it themselves? And the answer is simply if diffusion can't take place. So say if we need to move these particles here across to the other side, but we've already got a lot of particles, the same particles, these sodium ions, already here. So if we were to just leave everything to diffusion, then they'd all come back the other way, which is obviously not what we want. So we'll have other channel proteins like this that will be, say, closed at this end, and then we'll let these sodium ions throw in, and then we'll close this end, and kind of like a one-way ticket gate, these sodium ions, when they move through in the opposite direction to what they would have if we just left them to it. And that process, the opening and closing of these channels, takes energy, and that's why it's active transport as opposed to just a passive process. Is that all right?
Yeah.
Yeah. Cool. So quite a lot of marks. 9 marks for question 1. They'll do this often where they start off kind of simple. They're saying, right, we're talking about cells. This is an easy question on cells. And then you slowly get into a bit more detail as we go on. So question 2, start off with a 1 mark, define transpiration. Again, they give you lots of space, either to make you think you need to write more, or maybe just people with big handwriting, who knows. It's a really simple definition. I don't really know what else to say other than just be careful on your wording. Specifically, we're talking about water making its way to the pores in the leaf where it is evaporated. It's the loss of water from a plant. You can't get away with just saying, water evaporating off the leaves, because doesn't quite say what we're talking about. We're talking about water from inside the plant, as opposed to just resting on it. So just be a little bit careful with exactly how you're wording your definitions. I suppose I should be writing these little things I'm saying down. I should get used to this. I don't know why I changed red. I think I like red more than yellow. So part 2, we go on to talk about some environmental factors that affect the rate of transpiration. It's asking if you give 2 factors, so that's 2 marks. If you can just name 2 of them, doesn't matter if you don't know anything about them, that's 2 marks easy. And then a brief explanation as to how that factor has an effect. And there's all kinds of different things we could talk about here. Most obvious one that comes to mind would be temperature. Water is going to evaporate more quickly if it's a hot day, so we could have said temperature is our factor 1, and said what I just said there. Talk about how when temperature is, let's see if I can write this out. So when temperature is high, the rate of transpiration will also go up, and so a plant's response to that will probably be to close the stomatas on its leaves so that it doesn't lose all of its water. Because otherwise the plant will dry out, and that's obviously not what we want. Some other factors we could talk about. We talk about humidity would be one. Humidity kind of comes usually in with temperature. If it's a very humid day, which we're talking about, there's a lot of water in the air, we're not going to lose as much water through transpiration because there's already a lot of water in the air, so that gradient difference is not quite as high, so the water's not going to rush out as quickly as if it was bone dry, like out in the desert or something. We could also talk about this. I think there's a few more in here in the mark scheme. You can talk about wind as well. If it's a very windy day, it's going to carry the water off the leaves, and then that gives room for more water to come out, and we're going to have more water loss. And generally speaking, in any of these conditions, the plant's going to try and usually counteract that and work in the other way, so it doesn't lose all its water. Or if it's particularly humid, it might have too much water, which is also not necessarily a good thing, so it might want to open its pores and let more water out instead. And like I said, we could go on all day about different factors for transpiration, but we only need to name 2. Usually wouldn't expect them to ask you to talk about more than 3 at max. So rather than learn every single one, maybe pick 2 or 3 that you think, I can explain it easily, that makes sense, and just go with those. Is that all right? You good with that? Cool. Explain how hydrogen bonding is involved in the movement of water through the xylem. 3 marks, so a bit similar to the question we had before. Kind of a bit of a chemistry question, really. So we're talking about hydrogen bonds. How do we feel about understanding hydrogen bonding on a whole? Should we quickly go over what they're talking about so we know what we're explaining? Or you couldn't do this one? It's a tricky one? Okay. So when we're talking about hydrogen bonds, it's a shame it's not blue, so it would be good for water. But
I think that if you press on the color thing.
Oh yeah, look, we can change it to, so we can go, that's a bit pink. There we go, we can get a nice blue. Wonderful. So we're talking about water here. So we want to have an idea of the sort of structure of water. So these are it's a bit smaller, I should have done it a bit bigger. These are our hydrogens, and this is our oxygen, it's H2O. That's what we're talking about. Now, obviously in water you will have millions and millions of these molecules. A thing to know about what hydrogen bonding is it's not a bond in the same sense that this oxygen is bound to this hydrogen. It's more to think of it more like an interaction between the molecules themselves. And when you've got a lot of these lined up, you find that the charge on this molecule is not quite even, and you'll have this region around the oxygen be particularly negative, where it's bigger and it's attracting more of these electrons in, and these outer regions are more positive. So when you have a lot of these all crammed together, you find that they kind of stick, and you know that water is a little bit sticky, isn't it? It will, if you wash your hands in water, it will just rush off instantly. It kind of sticks around and that. And if you watch, say, raindrops on a window, you'll find that raindrops tend to want to converge together and sort of pool together. And that's this is why that happens. So I'll finish this diagram here. So all of our water molecules are going to kind of stick together like this. You see what's happening here? They're all kind of going to form this kind of a mesh, and it's not, as I say, it's not a strict bonding, like once they're in this formation they'll never lose, but they'll kind of prefer to be stuck like this than completely spread out. So that's what they're talking about here when they're asking about hydrogen bonding. That says, explain how this is involved in the movement through the xylem. So we do need to know a little bit about what they're talking about when they say the xylem here. Let's see if I move. There we go. So the xylem is it's basically a tube that the water moves through in a plant. It's the veins of the plant. It's how the water gets around. And there's two kind of interactions that we want to talk about. We want to talk about two things called cohesion and adhesion. These are the two, I could have just typed this, but I'm writing it now. These are the two keywords that they're looking for here. If you can explain these 2 points, then that's 2 marks. You've done most of the question. So cohesion, keep up being co, is talking about what we're describing here with the water molecules sticking to each other. And adhesion is talking about these water molecules sticking to the actual wall of the xylem. If you look at water in like a cup or in like a straw, you might see that rather than the water level just being completely flat, it actually kind of sticks up on the sides ever so slightly, like the edges of whatever it is. Like you can particularly see that in like a very thin tube like a straw, and that's because of adhesion. That's because these water molecules, like they stick to each other, are sticking to the edges of, in this case, the xylem. And that's what it's asking about here in this question. The reason that water can travel through the xylem so efficiently, and why water can get up from the roots of a plant all the way up through the stem, is because the water sticks to each other, so it will kind of drag the rest of it along. We're talking about water in very small quantities here, so I think the actual force of gravity putting it down isn't very high. It doesn't take a lot to push it upwards. So if you've got a very strong gradient of lots of water down here and none up here, and the water is sticky enough to hold on to the sides of this plant tubes, as we're saying, of the xylem, then we can move it up through the plant or to wherever in the plant it needs to go. So that's really as in depth as you need to get through here. It's not asking you to explain why hydrogen bonding is a thing in the first place, but it helps to have an understanding of what it is you're talking about when we're mentioning cohesive forces between water molecules and adhesion forces between the water and the side of the plant. So yeah, if you hit, say, those 2 points, it's a 3-mark question, but hydrogen bonding comes up a little bit later on in the paper as well. So any questions about that, or is that cool? Yeah? Cool. And so we're on to question 3. We've got a long snaky diagram, and the first question is to name the 2 types of covalent bond in this structure. So specifically, to point it out, we're looking at these bonds here, and we'll go like green. We're looking at the bonds here. Now, what are we actually looking at? It just says the structure of the enzyme, lysozyme. So it doesn't really tell us what we're looking at. All these letters are abbreviations of amino acids, and this is an amino acid chain. So what we've got to do is to be able to name what bond is keeping all these amino acids together and what bonds are holding the sort of secondary structure of this chain together. So we've got disulfide bonds here, clue being di to sulfide is the S there. That was the hint there. And these are peptide bonds, which is the bond in the process that creates amino acid chains. So those are the two that we're looking at here. Part 2 then says, explain the meaning of the term primary structure. So it's 4 different kinds of structures we're talking about. It's asking about the most simple of those. 1 mark, so we don't really have to go into any detail. We just have to say primary structure is, and the answer then is it's the order of these amino acids. So the order of these, all these abbreviations is important. If they were in a different order, this enzyme wouldn't work the way that it does. They might change this out for secondary structure or tertiary structure, which would be a completely different explanation. But for 1 mark, it's a simple. You just have to say it is this. It does say specifically to ignore reference to disulfide bonds, as we just pointed out, because that's not really, doesn't have anything to do with the actual order of these amino acids in the chain. So just beware not to think you need to include everything you just spoke about in part 1. When it's such a simple, just 1 mark, just remember your facts and just state just what needs to be said, rather than try and fill out all 3 lines. Joy, part B. Lysozyme hydrolyzes the beta-1,4 glycosidic bonds present in compounds found in bacterial cell walls. Very wordy question, isn't it? It's lovely. But part 1 is just asking if you know what the term hydrolysis is. If you have trouble remembering, bear in mind that any word that ends in this lysis part means to split up. So in terms of hydrolysis, we're talking about, let's just get the actual, here we go. So we're talking about breaking up a covalent bond with the addition of water, which is where this hydrolysis is breaking up with water. So that's a 1 mark, define that term. Suggest the type of biological molecule, which is the substrate for lysozyme. So when it's saying what the substrate for this enzyme is what does this enzyme act on? What's it made for? So it's not going to fall out of my pocket again. So you can say a few different things here. Specifically, it says that it hydrolyzes bonds present in compounds found in bacterial cell walls. This is the kind of hint they're giving you here when they're asking for the type of biological molecule. So it gives the answer peptidoglycan, which is the main component of bacterial cell walls. So not much more to say other than to be able to know. A bit of a tricky question. That might not be something you remember, but it is only a 1 mark. So that's pretty much it for that one. You could have also said, if you didn't happen to know specifically what this molecule was, you could have just said carbohydrates or like certain sugars like polysaccharides, and they would have accepted that as well. But yeah, they were looking for the molecule found here. Question on induced fit, which is a process involving enzymes. Is that something we want to look at in a bit of detail, maybe? Is that a bit tricky?
Yeah, maybe.
Okay. So when we're talking about enzymes, we're talking about things that speed up or facilitate reactions. Okay, so we've got 2 things that we want to combine into another thing, but the reaction is very slow, and maybe the one thing that we need is part of something else, and so we need something to help this reaction take place, and that's what we're using enzymes for. The way in which they work is a bit fluffy. There's kind of two theories on how they work. One is called Induced Fit, and the other is called Lock and Key. You might have heard those terms before. You should flick through a textbook. They're not incredibly different. They're kind of two explanations of the same thing happening. So if we start with our enzyme, in this case lysozyme, we'll say it looks a little something like this. Okay? This sticky out bit here probably looks a lot better than this under a microscope, but this part here is called the active site. And this is the part of the enzyme that's actually doing the work. How we explain how it works, we're talking about induced fit here. So the idea is that this pointy bit acts a bit like the end of a puzzle piece. So our substrate, in this case, this biological molecule that we just named, it doesn't really matter what it is. I'll do this in a different colour so it's a bit clear. Our substrate is going to be whatever shape with an active site which will complement the sort of puzzle piece on this enzyme that we've got here. Now, the induced fit theory says that the active site on the enzyme doesn't quite exactly match what we've got on the substrate. I'll put an S there for substrate so we know we don't get confused. So induced fit is saying that they don't exactly match up. It's not going to be lock and key like the other one we're talking about is where they just kind of fit and everything's happy and I don't know why I did that. Okay, see if I can neaten that up a bit. I was going to have to draw it again, aren't I? Yeah. So induced fit is saying that it's not as black and white as, oh, the enzymes just fit quite happily and then the just have to draw it a bit slower on it. I don't know why it doesn't A there, it's meant to be an E. So the enzyme is trying to say, oh, they fit happily and happily ever after. Maybe this breaks off of the big part of the molecule that's part of an reaction to take place. Wonderful. The induced fit is suggesting that instead of it just being, oh, they are completely complementary, they might be maybe not quite the same shape. So maybe on the enzyme, the end looks a bit like that. And when it comes into contact with our substrate, which is in green, when it comes into contact with it, what it does is it induces the fit and it will change. So the end might sort of sharpen a bit to then fit the substrate. So that's the key point here when they're asking about the mode of action that the enzyme uses. It's saying that the key points you're hitting here are that the active site on the I'll change that to an E, on the enzyme and the substrate aren't exactly complementary, but will be, where are we going? This is a very blurry camera.
Okay, you have to go there. Let's go. Ignore.
I think it crashed. I think that's what happened. Oh dear. Oh, has it got rid of all of our drawings? Oh no, it's just we've just been rewound, so all of my diagrams are gone. Oh, brilliant. This is we want this one, don't we? Yeah, okay. Are we good, I think? Yeah, alright. Well, that's brilliant. Well, this one, there we go. So, slightly cover. Do I need to draw that out, or do we kind of think we've got the gist of that now? Yeah, so, when we're talking about these fit mechanisms, either be it lock and key or induced fit, we're talking about this kind of puzzle piece idea of enzyme fitting a substrate. And this kind of idea of puzzle pieces kind of comes up a few times in a few other sort of mechanisms we'll talk about later. So that's about really as complicated as we need to make it. So we see, just to make sure we're hitting all four of our marks here, where are we? So 3b part 3. So yeah, so we're getting a point for saying that the shape is not completely complementary with the active site, so it's not quite the right puzzle piece. We're saying that on the enzyme, this puzzle piece point, this active site is kind of flexible, and when it enters this substrate, it's gonna change shape to kind of fit the mold, which means that they then do fit, and the and what's called an enzyme substrate complex is formed, which is when they're bound together, and we can have the reaction take place. So yeah, 4 marks, quite a long question. They don't get much longer than that in this paper. And then right at the end, they throw in a little quick explain the statement. State what is meant by the term extracellular. Again, the clue is kind of in the name. It's not really it's not like it's a Latin word. We have to remember we've got 2 parts to it. We've got the word extra. Usually, if there's an E and an X, it means outside. Exothermic reactions give out energy. So we know that that's outside, and then obviously we've got the term cellular. So we can kind of put 2 and 2 together and figure out that we're just talking about things that happen outside the cell. That's basically it. We don't need to say much more than that. That's about it for that. We have part D. I had to double take on this question when I looked at it,
Okay.
but it's not as difficult as it seems, fortunately. So we've given a graph here plotting the rate of reaction to the substrate concentration. So how much stuff we've got and how quickly the reaction is taking place. Just put that in layman's terms. Part 1 asks to state the lowest substrate concentration to give the maximum rate of reaction. So to kind of reword that, I mean, how did you guys find this question? Was it a little bit weird, or was it all right?
For me, it was quite easy.
Quite easy. Did we struggle on this one at all, or should we go through it like quite slowly, or are we good? We good?
No.
Oh no, we're not good. That's all right. So
These are emersomes. You said you.
yeah, it's tricky stuff. It is. So we're asked to state the lowest substrate concentration, which is on this x-axis here, to give the maximum rate of reaction, which is on this side here. So we're looking for the maximum rate of reaction. So we're looking for basically the highest point along this axis that we can get. So if we were to, I'm not going to try and draw a straight line. Or is there a straight line? It's a straight line tool, isn't there? Oh, where have they gone? They were like...
Yeah, that one.
Oh, it's there. Okay. So, perfect. So we find the highest point here, which is it's going to reach its peak at about this point here. So we'll follow that down. Oh, look, that's perfectly straight. And read off whereabouts that is. They give you a little bit of leeway. Maybe you said, oh, it doesn't quite reach the top until 3. So you can, the given answer is 2.9, which is about here, but they say you can have anywhere from about here to up to 3. So if you're somewhere in that area, you're fine. So that's all you need to give, and you're just reading it out of the graph. Where's the highest point? Follow it down. That's Pmax. That's what we need to give there. It then asks you to determine the, I don't even want to try and pronounce it, the Michaelis-Menten constant. Michaelis. I'm not entirely sure.
Is it German?
It might be. It looks like chemistry stuff is quite often named after German people. Yeah. I find this was never around when I did biology and chemistry, so this stumped me a little bit at first. But what it basically there's not actually any calculation involved, which is nice. Well, there is, but it's very minor.
You just have to find half.
Basically, yeah. So when it says Km, it's asking for the rate of reaction at half of Vmax, the amount of substrate we're using at half of Vmax. Found we were using Vmax just a second ago. So it's not the number we gave. We're looking at the highest number here, which is let's use like a blue line. I like blue. So we'll read that off the. Well, apparently it's still red. Okay, that's fine. So that's 3.2.
You can make it blue if you want.
Yeah, can I do it? Well, there we go. Oh, can't we see? Maybe yellow. No, easy yellow. So we find our Vmax, and we want to know the substrate concentration at half of that. So 3.2 divided by 2 is 1.6. So we'll go up to our 1.6. Where's our line? Nice easy numbers. There's not a lot of decimals or anything involved. It's on this cross here. 1. So our answer is 1. And that's it. There's no need to do lots of calculations or anything involved. There is an equation involving the constant, but we're not asked about that here. So
I've got a question about what the unit is really.
Yeah.
Isn't it substrate? Yeah, concentration like amount of moles per like volume. So,
yeah.
so what does m mole mean really?
In this context, I hate to say I'm not sure, but I think I think it might just be millimoles. That might be what we're talking about here. So we've got micromoles per minute up here, which is how much of it is being used up. So I would assume this is just talking about millimoles. So we're talking about it in quite small quantities.
Okay. It's just because concentrations are normally
they're using
moles and volume.
Moles per dm cubed.
Yeah.
So fortunately, this is AS biology, so we're not too worried about concentrations. Part E says, all right, so we're asked to draw a curve in the presence of a competitive inhibitor. So we're seeing if we know the difference between a competitive inhibitor and a non-competitive inhibitor. There's two types of lines we draw. It doesn't have to be particularly well drawn. It can, as long as there's kind of 2 points that we want to hit. So what do we mean when we say a competitive inhibitor? We were just talking about active sites on enzymes, weren't we? About this kind of puzzle fit. So we've got, during this reaction that's taking place, we've got our all of our substrate, which is being used in the reaction, and we've got our enzymes that come along and catalyze it, that help it move along like this. A competitive inhibitor is going to be another substance that's introduced that competes for this space here, for this puzzle piece space. So if we've got all this and there's no inhibitor whatsoever, we're going to follow this black line here. It's going to happen quite steadily until it kind of reaches the max where we're not getting any more out of this reaction. If we've suddenly thrown in lots of these competitive inhibitors that are kind of stealing the spaces for these, either these enzymes, we might be talking about that. So they might be using up the enzymes, so the whole process is going slower, or the substrate is being used up and not actually used in the reaction that's taking place. We're going to have a much slower reaction because everything has been slowed down by this inhibitor, which is competing for space. But what's important to note is that although we're going to have a much slower reaction taking place, eventually, as we go along this axis and you add more and more substrate, eventually it will use up all of the substrate and we will reach our maximum rate of reaction eventually. It's just going to take us longer to get there. So our curve that we draw is going to look a lot neater than this maybe, but something like that. And they're going to meet, that's awful, isn't it? They're going to meet at the same point, but it will happen a lot slower, and so you'll have a much lower gradient, which is what they're looking for. They're looking just for a single graph line with a lower gradient that reaches or approaches a plateau at the top. Answer would have been different if we were talking about non-competitive inhibitors, which, we don't have to go into too much detail now, but our line would look a lot different because this whole thing going on is not working the same way, and we're changing our enzymes and our substrates so that the reaction can't take place. But we won't go too much into that because it's not completely relevant for the question. So Is that clear? You're all right with that question? Cool. Question 4. This was my least favourite question in the paper.
Yes.
My personal opinion. We're talking about cholera. Lovely stuff. Name the bacterium that causes cholera. There's not really much to say, just knowing the Latin name for it. I've always struggled learning Latin names. I'm not really sure what to say other than try and commit them to memory, to be honest. But it's only 1 mark, so we're not losing big points here for not remembering that. Describe how cholera is transmitted from an infected person to an uninfected person. 2 marks. So we're seeing if we know how cholera is transmitted for a start, and specifically how it might travel from an infected person to someone who doesn't have it. So cholera is often found in water. It's transmitted by water. That's where the bacterium lives. But we can't just say, oh, from polluted water, because we're asking how it might travel from someone who has a disease to someone who doesn't. So we get to talking about lots of lovely stuff like feces, passed through in stools and in sewage, where it can infect water supplies that are then drank by other people later on. So that's how someone might move the bacteria from themselves into the environment, and then that might lead to contaminating food or water that the uninfected person may then drink, and then there you go, you can now have the disease. Lovely stuff. So you could have, suggested a few different ways. You could have, talked about contaminated food or like water supplies, or using a fork that's got some cholera on it, or or whatever. But that's what we're talking about here. So lovely stuff to talk about. But part 3 then says suggest and explain why cholera outbreaks are common after natural disasters. So suggest and explain. We're looking at 2 marks. So we're looking for something that might happen during a natural disaster, and why that thing that happened would cause this outbreak. So we can suggest any number of things, really. There's a big list. Maybe there's an earthquake and it damages the water, like pipe systems, so sewage and clean water get mixed up, and cholera is spread that way. We could reference to flies being suddenly exposed to a lot of sewage that was previously packed away nicely and is suddenly now out in the open. It even goes on to talk about things like needing to share your neighbour's cooking pots. Maybe yours got blown out the window by a hurricane, and you need to borrow your neighbour's, and they don't wash up quite so well. So there's all kinds of different things you can say. Not very many wrong answers, really, as long as you can explain why that might be a bad thing. You can talk about any number of things. We could be here all day naming them all, but you only need to name 1 or 2 to get your max marks. Or if you give 1 and explain it well, then you're fine. So that's that, really. And then we talk about bacteria in a bit more detail in part B. So the bacteria that cause cholera can become resistant to antibiotics by substitution mutation. A substitution mutation occurs when one nucleotide in a DNA sequence is replaced by a different nucleotide. Quite a wordy question. Explain how this substitution mutation could result in a change in the amino acid sequence of a polypeptide. So it's kind of giving you everything you need in the question, because it tells you that we're talking about substitution mutations and a change in the amino acid sequence. And it doesn't really say explicitly, but it's saying that it's telling you when this happens, we get a change in the amino acid sequence. Why does that happen? So we need to understand that DNA sequences, when we're talking about our sequence of base pairs, so along this helix of DNA, we might have a C, T, T, G, A, go on, it goes on for forever and ever. every 3 that we get codes for a certain amino acid. So this combination makes 1 amino acid. If it's a G in the middle, then it's a different amino acid. And it tells us that a substitution mutation occurs when a nucleotide in the sequence is replaced by a different one. So this long sequence, if this bit here is suddenly cut out and replaced by a completely different set of numbers, it's meant to be like a G, maybe it's G double T. You'll suddenly, when you put this long list of amino acids together, it's going to be different, and your chain then it's a bit of a butterfly effect, that this chain, because it's now made up of a slightly different sequence of amino acids, might fold into a different shape later on, which means it doesn't perform the job it's supposed to do as a protein later on. So it's a bit of a butterfly effect, but it gives you a couple of kind of hints and helping hands in the question, because it tells you exactly what is going on, and it kind of hints at the effect. You just kind of need to put 2 and 2 together and explain why, if that makes sense. Yeah, we good? So part 2 kind of follows on from that, which says there's a lot of reading going on in this question. This is, I'm not even sure it's worth quite a few marks, 13 marks total, so it's quite a big chunk of time, I suppose, spent. The antibiotic nalidixic acid acts as an inhibitor of an enzyme involved in DNA replication in the bacteria that causes cholera. The gene GYRA codes for this enzyme. A substitution mutation in this gene results in resistance to antibiotic nalidixic acid. Lots of words thrown in. It's a bit of a wordy jumble that kind of throws us off a bit. But all the question's asking here is how a change in the amino acid sequence of the enzyme results in antibiotic resistance. So we kind of started to talk about this in part 1. And we sort of started to explain it a second ago when we were talking about this chain of amino acids being different, and for that reason it folds into a different shape, which means that its secondary or tertiary structure is different, which could mean that, or suddenly this enzyme can't do its job, because it's meant to be this shaped puzzle piece down the line, but it folded into the wrong shape, and now it doesn't fit anymore, and so it doesn't work.
The structure is
now changed and is now different. Yeah. So the binding site for this antibiotic, which is supposed to attack this this disease, suddenly can't because it's the wrong shape. And so where are we? This antibiotic resistance has come up because this enzyme no longer works and can't function as it should previously. So 2 marks, so you don't need to include absolutely everything. You can get a mark from just saying that because this polypeptide chain is now different, the tertiary structure has folded differently. That's 1 mark. Easy, that's halfway there. To then just go on and say that the enzyme's binding site is now different because it's changed shape, so it can't bind with the nalidixic acid. There you go. That's it. There's your 2 marks. That's all we need to say on that one. Moving on, part C. Some of the strains of bacteria that cause cholera are resistant to more than one antibiotic, multiple resistance. Explain the consequences of this. So not really a biology question, but long 4-mark question to talk about more sort of practical reasons about why this is an issue. So it's another one of those questions where, a bit like the one we talked about with natural disasters, there's a whole load of things we could talk about. You could say that, we can go through the list and pick out a few. If people are still infected because there aren't any antibiotics that work where they're from, and they go travelling on holiday somewhere, you've suddenly got a risk of more people being infected. The longer someone isn't treated, the longer the more risk there is of it being transferred to a lot of people. And so on. We can talk about the fact that it's all of a sudden it's more expensive now because you've got to spend money developing new antibiotics. So this is strain on everyone's pockets. Everyone's working overtime. We can talk about the fact that hospitals are now open longer because we're spending more time treating people because we haven't just got a source of antibiotics to spare to give to people, and just generally the cost of having to research suddenly these and find these new alternative treatments. So really, there's a whole plethora of different things that you can talk about as long as you don't repeat yourself and mention the same thing twice in different words. I don't really see too much of a ways you could be tripped up here. So, and that's a long 13-mark question. So most of the marks can come from there's a 2, there's a 2, there's a 2. There's a lot of writing to be done here in question 4. But how do we feel? Generally okay with that? Or anything you want to go back over? Or are we okay? You good? Cool. Question 5 then, which I think is the last question. There's only
There's 6.
Oh, there's a 6. Okay. Okay. So monoclonal antibodies have been used in treatment of some non-infectious diseases. They can be designed to bind to a protein on disease cells, so causing their destruction by the cells of the person's immune system. Name the part of an antibody molecule that will bind to a protein on disease cells. So we're talking about the immune system and the functions of the immune system here. So the named part is, make sure, we're talking about the binding site of the antigen, so the antigen binding site, or the variable region. There's 1 mark there, not really too much to say, I don't think. But part 2 goes on to ask to suggest how the binding of this antibody to the disease cells actually causes their destruction. It doesn't just say full stop, oh, how does it cause their destruction? It says by the cells of the person's immune system. So that kind of gives us a bit of a hint, because this is 4 marks. We can get 1 mark already just by having read the question, because we know that if they're being destroyed by a person's immune system, we have stimulated an immune response, and that's 1 mark already. So we kind of want to explain some of the finer details of how the immune system works and how it tackles diseases in general. So if we understand that, generally speaking, our own antibodies that we have will recognize antigens on pathogens. So say it's a cold virus or something like that. And the cold virus looks like, I don't know, like this. It's got these things sticking out on the end. And we have our antibodies come along that recognize this site here, and they bind onto them, something like that. So they clump onto them, and this happens a number of times, and you get all these disease pathogens get clumped together, where they can then be swallowed by a big white blood cell and destroyed that way. And there you go. You slowly, over time, get rid of the disease from your system, and you're fine. So the same sort of thing's happening here, but we're talking about our own disease cells. Usually, the immune system isn't going to bother touching your own cells, because if it did, we'd eat ourselves from the inside out, and that'd be terrible. So the main sort of reason we use these monoclonal antibodies is that your immune system recognizes them as they're not ours. That's not our stuff, so it must be bad. So we need to get rid of those. And we're kind of exploiting this system by having these antibodies come along and do what our antibodies do, but instead of latching onto a pathogen that's coming from the outside, they're latching onto our disease cells instead. So if you've got a particularly nasty cell that's you need to get rid of because it's diseased, and you have these monoclonal antibodies come along and pick them up. Diagram's not very good, I apologise, but you get the idea. Your immune system suddenly says, oh, hang on, this thing here, that's not ours. We need to get rid of that. And so the same sort of thing can happen. They can all come together, and when your big white blood cell comes along and decides to eat the whole thing, it's eating these disease cells at the same time and destroying them. So it's a very sort of clever way of kind of exploiting your body's sort of natural way. Your body doesn't really know what's going on. It just kind of thinks it's doing its job, but these antibodies are kind of sweeping up the disease cells we don't want along with it to get rid of them. I mean, this is a 4-mark question, so you don't need to hit every single point. But if you can kind of get 4 of the key steps in there and make sure that you end on saying that how these disease cells are then destroyed by our white blood cells, by our phagocytes, then we're okay. Generally speaking, in a long sort of question like this, if it's only so many marks, like only 4 marks, what they're saying is you want to get 4 key points in there, but you want to kind of get the start to finish. You don't just want to stop halfway through the process and saying, oh, I've done 4. You want to at least say, it starts like this and ends like this, and just have enough points in the middle to count up however many marks that we're aiming for. Is that okay? Are we good with that? Cool. I think that's it for... Oh no, there's part B. Okay, so explain the term autoimmune disease. We're not bothered about the fact that this is an autoimmune disease. That whole line is irrelevant because it's just asking us this. So that happens quite often. Sometimes it's just a line. Sometimes you might have a paragraph that you don't need because if you read the question, might just be asking you to explain something you remember. So, and in this case, an autoimmune disease is where earlier on where we were saying how the body recognises that's ours, that's not ours. It's got that mixed up, and it might think that some of your healthy cells aren't meant to be there, and it will attack those. And that's what autoimmune disease is. It's only a 1-mark question, so we're just looking for a simple explanation. You really need 1 or 2 lines. You don't need to fill out the whole thing. You can just say that. You could just, like it says in the mark scheme, you can just say a failure to distinguish antigens from our own cells and from pathogens. That's it. There's your 1 mark. Short question, but if you, I suppose, if you miss out on explaining part 2, you miss out on most of the marks in that question. So there you go. It's only 6, and then this is the final question. It looks like we might make really good time, even though we started a bit late. We're given a table. Place a tick where a feature is present, and a cross where a feature is absent. So we're really just seeing if we remember what each of these things are. We know we're talking about features of the human gas exchange system. Specifically here, although it doesn't say, we're talking about lungs. These are all different components of the lungs. Our trachea is our sort of main windpipe, as we usually call it. So it's going to be our main sort of structure. So we know it's probably going to need cartilage because it's the biggest thing. We don't want that getting squished closed when we lay a funny way or something, so it needs to be held open the whole time, so it's going to have cartilage. We know it's going to have cilia because that's where all the mucus is going to come out when we cough up horrible amounts of phlegm and stuff. That's how it gets into our throat to begin with, because these cilia are working to move all of this mucus up. And we know that all of these are going to need elastic fibres because of the very basic function of the lungs. They expand and then they contract as we breathe. So we know all of it's going to need to have that kind of give. And then we kind of move down into the bronchioles, which is where our lungs start to kind of branch off. As you know, if you've ever seen like an X-ray, well not an X-ray, but like a scan of someone's lungs, you know, it looks like a big spider web. So, and as we get smaller, we know we're not going to need as much cartilage anymore because they're small enough to that the force of the air going through it is going to keep them open. So we don't need to worry about that. Cilia, we know it's going to have cilia because, as we just said, we're trying to move the mucus out of these areas, and we've already said they're all going to need elastic fibres. And then the final part is the alveoli, which is the tiny, tiny little air sacs where the actual gas exchange takes place, so where all the blood vessels run over and where the oxygen can move through. So we know we're definitely not going to have any cartilage, there's not enough room for it. And we won't have any cilia there either because its only function is gas exchange. That's off across. There we go. But all right, so it's just basic parts of the lungs, pretty simple. 3 marks though, so if you can remember all of that, you can pick up, that might be 2 defined questions that you couldn't get earlier on, you can make up for now, because, it's just an easy tick across, so nice to be able to get these questions all in one, because they're usually worth a few marks. Part B, smoking causes changes to the structure of the lining of the bronchi that makes smokers more likely to be infected by bacteria. Describe these changes and explain how this leads to an increased risk of bacterial infection. So inferring some knowledge here about the lining of the bronchi and what cells we find there. So 3 marks as well, so a little bit in depth, but what we're talking about here is what we were checking for here with the cilia. Smoking does all kinds of things to your lungs, but one of the things it does is it damages these cilia. And the cilia's role is to, kind of like a tennis bat, is flick all of the mucus in your lungs out so you can cough it out. Why does it do that? It does that because mucus is, as gross as it is, it's the important part because it catches all the bacteria. When you're breathing in stuff, you don't know what you're breathing in. It could be nice, clean, fresh air, or it could be full of loads of horrible bacteria that if that just sat around in your lungs, that's obviously going to cause you a lot of harm. So the mucus is there to catch all of that, and then the cilia kind of bat it away, like a big net almost. So if you're smoking and your cilia are suddenly damaged, that mucus is still going to catch bacteria, but it's not going to go anywhere, or it's going to not be removed as easily. That's why people who smoke cough a lot, because all this mucus is just being stuck in all parts of the lungs, so they're having to heave it up manually. And so our increased risk of bacterial infection comes from simply that bacteria is hanging around for longer, and so it has more of a chance to embed itself and stick around and multiply and cause a bacterial infection. So those are the sort of main points we want to pick up on there. That clear? We good with that? Yeah. Part C, it's like our second to last question here. Talking about hemoglobin. We spoke, I think there was a bit about that in the paper yesterday. Hemoglobin binds oxygen in the lungs to form oxyhemoglobin. Describe what causes the oxygen to be released from the oxyhemoglobin as it flows through the respiring tissues. So we know that hemoglobin is used to move oxygen out of the body as we breathe it in. It gets bound to the hemoglobin and then carried to where it's needed. How do we know when it's needed, and how does it get off the hemoglobin? How does it get off that roller coaster is what it's asking. So 4 marks. Just make sure I get them all mixed up. Yeah, there we go. So we know that when the oxygen does get to where it needs to go, it's going to get used up in respiration, and it's going to be used to create energy, and our waste products are going to come back as CO2. So we know wherever it's going, there's not going to be as much oxygen there. So that partial pressure of oxygen is going to be more leaning towards the hemoglobin. So there's going to be that want for the oxygen to diffuse over from the hemoglobin to the cells where it's being taken. And likewise, the carbon dioxide that's already there is going to be much higher concentration in these cells that have just been working compared to the hemoglobin. So there's going to be a kind of sort of seesaw kind of swap going around. And you might remember from yesterday, we knew that oxygen and carbon dioxide bind to different places on the hemoglobin. So before the oxygen's even left, the carbon dioxide can come along and start binding to the hemoglobin. And when it does that, it releases the oxygen in turn because of the H+ ions that get released when that takes place, and when it forms this kind of complex between the hemoglobin and the carbon dioxide. So it's that. So what we're saying here is that when the carbon dioxide comes in and binds with the hemoglobin, that kind of almost like pushes the oxygen out of its place, because the hemoglobin is changing shape and it's binding to this carbon dioxide, and the oxygen is then being released, and the reason it moves through is because of this pressure. There's no oxygen here, we'll just use it all up. So it's going to diffuse along that partial pressure gradient. Yeah, and that's those 4 marks there. So mentioning the fact that the oxygen gets used up in respiration, so that there's a decrease in that partial pressure, as we just talked about. Yeah, and then talking about its affinity for oxygen when the carbon dioxide comes in, because of the other pressure gradient. Yeah, is that all right? Are we okay with that one? It's a bit of a wordy sort of question there, but those are sort of the main points. And then, where's the pen going? Okay. One final question to finish off. It's just a simple 1 mark. Explain why red blood cells are in blood, but not in tissue fluid. Simple answer being that they're too big. Obviously, don't just write they're too big in the exam. You probably might appreciate that. But specifically, they're too big to pass through the walls of the capillary. Smaller things can get through, but these guys can't because they're too big. And that's the reason they're so big, is so they don't accidentally leak out everywhere, because they're needed in the blood for carrying oxygen. So, and that's that. Not too bad, really, is it? It's all right. Is there any questions you wanted to quickly ask? Any points that we maybe went over too quickly, or we think we're okay?
Are you okay with?
What is the hydrophobic?
Hydrophobic. So we're going back to, I think this is that diagram there. Oh no, it's here. Are we talking about this on the hydrophobic heads of the membrane? Is that what we're talking about here?
Yeah.
Okay, so if we look closely at this, it's called a phospholipid bilayer, because I'll just quickly get to it. I know it's half past, but we'll just get this last point. Because it's made up of a lot of phospholipids, which you can kind of imagine as a head and a tail. We call the membrane hydrophobic, hydrophobic meaning that it doesn't like water, because these heads repel water, basically. The reason they repel water is because if you've got a lot of these lined up like this, on the inside of the cell, we've got our stuff floating around on the inside of the cell here. It's not all then going to leak out of the membrane because it repels water in such a way.
Isn't it the tails that repel water? Or is it that?
I'm reasonably sure that it's this way around, that the tails are hydrophilic. Because if it was the other way around, then water would just be going in and out. Like all the stuff in the cell would just be leaking out all the time. Check the notes anyway. If I'm wrong, let me know. But I'm really certain. Is that right then? Yeah. Cool. I don't know how I go about turning this off. Yeah, there's anything. If I've ever said anything that you think, oh, hang on, I don't think that's right, do just stop me and say, hang on a minute, call me out on it, because as we're going through it all, there might be something I say wrong or something that maybe I've forgotten. I'm not perfect by any means, so just let me know. Or if maybe you notice it like way later on, just drop me a message or whatever, and the next time I'll make sure we go back and correct it. We'll all get there in the end. But so just press the stop button, do I? Is that right?
Yeah. Right.
I'll stop this live thing. Or is this...